So thank you for joining us. This is a webinar brought to you by Millrock and also from Baxter. That's where I work. My name is Greg Sacha. I'm a senior research scientist at Baxter Healthcare. And I'm at a site located in Bloomington, Indiana. That's our contract research and development and contract manufacturing arm of Baxter. So our group is, our research and development team is actually located on the manufacturing site. It gives us a bit of an advantage because we know what the end in mind should look like in manufacturing. And then we also do get called in when there are challenges that occur. And you know, during manufacturing, there's always challenges that occur. So it's a good learning opportunity for us, but it also helps the manufacturing team solve some challenges. Now, we also have a sister site in Halle, Germany, and their expertise lies in chemotherapeutic agents and highly potent materials. So they're working exclusively with isolators. And therefore, any of the antibody-drug conjugates that we develop here in our lab, they're transferred to our site in Halle. But what we'll do is we'll speak about the development of these antibody-drug conjugates. From now on, I'll just call them ADCs. And we'll look at them as a molecule and then also from a development standpoint. And just by the way, I should be used to virtual webinars and virtual short courses by now, but I find them a challenge because I don't get to see the audience, and I really enjoy being able to see you so that I know that I'm not boring you and so that I know when you have a question. So in that case, please stop me at any time. You can either raise your voice or we have the chat open, and Brian and I will both be paying attention to the chat. So if you have questions during the presentation, please state them. I'm sure if you have a question, others do as well. And it makes it a little more fun and interesting. So let's get started. The discussion points that we'll have are first, we'll look at just the ADC, their components and their mechanisms of action. So I was speaking with Kian Thompson and Brian about a month ago or so, and they had a lot of questions about ADCs, and they were interested in a webinar such as this, where we can at least introduce them, explain a little bit about what they are, and then also include our formulation and process development portion, where I'll tend to focus on some of the actual case studies from ADCs that we've worked on. Let's start by considering current cancer treatment options. They differ depending on the type of cancer, but solid tumors are typically treated with multiple methods. First, surgery, if possible. If you can use it to remove the majority of the tumor. There are even diagnostic agents out there now that can help highlight the tumor's edges around healthy tissue so that the surgeon can follow those highlighted areas and remove all of the tumor cell. Other methods are administering chemotherapeutic agents and then also radiation. So radiation is targeted as much as possible to that tumor area, but by and large, we still hit healthy tissue. So there's still healthy tissue that's deeply affected and can result in a lot of scarring. Chemotherapeutic agents, the small molecules that we currently have, they vary widely, but we usually have to infuse quite a lot of them on a frequent basis. And we infuse so much so that these small molecules can hit their intended areas, wherever the tumor lies. The big challenge there is when you're giving these large doses, they also affect healthy tissue. So the results here are severe side effects. The patients will lose their hair, they have nausea, fatigue. And this fatigue and nausea often require treatment with additional medications. So it becomes a very tough situation for the patients. These medications are most often given by injection or infusion, but there are oral medications available as well as topical, especially for skin melanomas. Some examples of these chemotherapeutic agents, and by and far this is not all encompassing. There are, I'll just name a few since you may have heard of them, you may have even worked with them. They've been around for a long time, such as antimetabolites. There's ones that are known as cytarabine and gemcitabine. They've been around for a long time. Alkylating agents such as the platinum-based agents like cisplatin and carboplatin, those are also well known and been around a long time, like nitrosoureas, carmustine. And there are even like the vinca alkaloids, vincristine, vinblastine. If you're a gardener or anything, you may have heard of these vinca vines that we can plant during the summer in the areas that where I live. And those vinca vines are exactly where these vinca alkaloids come from. So now let's consider an entirely different approach. Rather than inundating the body with these toxic materials, we can drastically reduce the number of chemotherapeutic molecules that we give to a patient if we can incorporate them onto an ADC, an antibody-drug conjugate. These antibody-drug conjugates are 3 molecular entities. So you have your monoclonal antibody, which in this diagram, this picture, that's that blue area, that blue Y that you're looking at. And typically, we build antibodies antigens. And so what is nice is that these antibodies can be chosen so that they will preferentially go to cells that are expressing a certain protein or chemical signal, such as a tissue factor. On these antibodies, since our bodies already know how to work with these, we can attach a chemical linker system. That's that yellow line right there, making it very simple looking. They're actually very complex. And those hold individual molecules of cytotoxic agent. So maybe 4 to 6 or 4 to 8 individual molecules per monoclonal antibody. That's a drastic reduction in the number of those cytotoxic molecules given to a person. So some desirable properties of these ADC components. Well, the antibody, we would like it to specifically target a certain cell that's expressing or overexpressing a certain molecule. So we hope these cancer cells are only expressing that target that the antibody is going to, or they could be expressing them. We want this so that the antibody drug conjugate has limited effect on healthy tissue. We like to have strong target binding affinity. The cytotoxic agent, or it's often known as the payload or the warhead that's attached to that antibody, we would like it to stay firmly attached to that antibody until it reaches its desired point or target. This allows us to provide very low doses, but still kill those tumor cells. Then the linker system. That linker system, we would like it to remain firmly attached to the antibody until it reaches its desired destination. The older linker systems, when this technology is first developed, tend to not link as well, and you'd have free linker and chemotherapeutic agent floating around. You don't see that as much anymore. These linker systems are firmly attached, and they're fairly stable when circulating through the body. And so we want that so that they deliver that payload only to the tumor cell, preferentially. These linker systems, they're both known as cleavable and non-cleavable. So here's just another example of the ADC platform. Here's your antibody in that blue and red, and this large linker system accompanied by the warhead. And so we could attach specifically 4 or 6 individual molecules to those antibodies. The linker systems, as I mentioned, they can be cleavable or non-cleavable. So we want these to be stably attached so that they don't release just anywhere within the body. They must maintain that conjugate as an inactive substance until it reaches its target. Non-cleavable linkers, these ADCs, are internalized into the cell where that antibody is then degraded, and that's what releases the warhead. Cleavable linkers, these linkers will cleave depending on certain environmental conditions, let's say. What's the pH? Perhaps a redox potential. They could be acid sensitive or be sensitive to lysosomal proteases, which will cleave that linker once it reaches its desired location. Now here's just an example of the marketed ADCs. There are 5 of them, at least that I am aware of. And you'll see on the left side of the table, you'll see the different ADCs. The next column is the antibody. I placed either humanized or chimeric here. In general, they all use a type of IgG, typically IgG1 or IgG4. Then currently there's cleavable and non-cleavable linkers that are marketed. And then each of them are associated with the payload. There are also a couple more that are still in development. Well, actually they're outside of development. We developed them here at our site. I have patents on the formulations for 2 of them, both here in the US and in Europe. But we transferred those to our site in Halle, where they're currently making clinical supplies. All 5 of these products that are currently marketed and the ones that we transferred to Halle, they are lyophilized. So that tells us something about them. That means they're not solution stable. So even they may be stable at very low temperatures, but what we've seen, what we've all been aware of with the pandemic, is that this cold chain storage is a huge challenge. Even if you can ship it cold chain and have it remain cold, the facilities that you're shipping to may not be able to have that type of low temperature storage. So in order to get around those incompatibilities with unstable solutions or low temperature storage, we could look at freeze drying them to extend their stability. So what makes an optimal ADC drug product formulation? Well, first, once we formulate it and we process it, that linker system must still remain stable. Again, just to mention, older linker systems, which are still available, they tend to not stick as well. And what that means is that when working with those older linker systems, you would have to work in treat it as a category 4 compound per safe bridge. And that means working with them in isolators. Otherwise, you risk the health of the people working with them. The current linkers that have very good stability, we can work with them as a category 3 compound and work with them in our laboratory, still taking appropriate precautions, just like we do with any other chemical, but we don't have to work with it only in an isolator. We want the monoclonal antibody to be stable. So throughout our studies, we're looking at potential for aggregation, or potential for fragmentation or deamination. And the way to improve this stability, this long-term stability, is often through lyophilization. So this is just a quick diagram showing you an overview of our process, or anybody's process really, we have to have good analytical methods in place to, in particular, stability indicating analytical methods. Where we usually start is with our formulation studies is solution stability studies. We want to determine what's the best buffer, what's the dependence on pH, is there a dependence on buffer concentration that would make a difference for the stability of this molecule. That will then provide us the basic data where we can start building our formulation for lyo development processes. And throughout all of these studies, we're working in areas that we expect some failure. We want to know where our product will fail so that we can stay away from those areas. If we can design a lot of studies where we never see any failure, we won't know if we're right on the edge of failure or not. So it's important to be able to test that. And lyo development, we also test for failure. I'll show you some slides later on. And then we conduct thorough secondary drying studies where we examine the effect of residual moisture on the stability of the product. And we'll provide you some case studies on that as well. So our analytical toolbox, all of these methods are extremely important, but there are a few of them that are more important than others at early stages. So for example, we may look at concentration, but I've never seen concentration change and does not really represent a great stability indicating method. What we want to know is, do we see fragmentation? Do we see aggregation? Do we see anything that would relate to this not being stable over the long term? So that I'll show you a slide next that would include that. So part of aggregation would be SEC, DLS, dynamic light scattering. Sometimes those are really good to use both at the same time because DLS uses very little solution. So it's almost like getting some data for free. The DAR-HIC, this drug antibody ratio with hydrophobic interaction chromatography, we use that to determine if we have our linker systems still in place. And as I mentioned, with the newer linker systems, we never see any changes with that. So we might include that particular assay, perhaps on long-term stability, just so we have that data. Our stability indicating methods, though, the most common, ICE, we may look at DAR or free drug over time, but it depends on the linker system. So really what we focus on is SEC and ICE during the early studies. We find those tell us a lot more information. Here's an example of free drug, and this is solution and lyophilized samples stored at 50 degrees C. We don't often store our solution samples at 50 degrees C, but for the next few examples, for some reason we did on these studies, and I don't remember the exact reason why. When we do, we store at 50 degrees C so that we hopefully can get some data quicker and maybe provide us some idea of what may come when we store it for longer term. It's frequent during our early studies that we'll store freeze-dried samples at 50 C for 1 and 2 weeks, but also store them at 25 C and 40 C for 2 and 4 months. So this is an example of monitoring free drug. Rarely have we ever seen free drug available in the formulations when we're using these newer linker systems. Here's also an example of the drug antibody ratio. Again, even under stressed conditions, I'm showing you here 40 C for 2 weeks, but we have data for 40 C for up to 6 or 12 months, maybe 2 years, where you do not see any changes in the drug antibody ratio. So it's usually just included as a check. ICE, however, we do use. We use this because we do see changes over time that may show us different charge isoforms that develop due to the storage conditions, but also due to our formulation conditions. This is just an example. This is a graph at T0, and then one after 1 week at 50C. And we see a large change. And so that tells us this will be an important method when we're working with needing a stability indicating method. SEC, size exclusion chromatography, we also use. We monitor soluble aggregates, and we do see changes over time. And I'll show you some examples, some statistical analyses that we've used where we use SEC and ICE to determine which direction we should go. So now let's take a look at the components of a formulation for a lyophilized antibody-drug conjugate. These are some common formulation excipients. I wouldn't use all of them, but some common ones are listed and why we would use them. For example, pH control or stabilization of the lyophilized solid, or providing bulk. But again, we hope that all our excipients that we're using are inert and they should not interact. However, they do. They do bring some interactions. And here's some examples. So one example, this isn't really an interaction, but sodium chloride. If we need to adjust the tonicity, we would only adjust tonicity if our route of administration is direct injection or into the muscle or under the skin, subcutaneous. If it's going to be administered as an infusion, we don't really need to worry about adjusting tonicity for that formulation. But if we choose to, and we're lyophilizing, we try to stay away from sodium chloride. Salts definitely decrease your critical product temperatures. So they lower them to temperatures that make it difficult to freeze or difficult to dry. So it's better to adjust tonicity with mannitol or glycine. We may include an ionic surfactant, and that's to inhibit aggregates. I'll explain a little bit more about that later. Buffers. Some buffers that we try to stay away from are phosphates, mainly because they can bring along with them some metals. They're known as types of chelators. There are, in those lists of antibody-drug conjugates that are marketed, they use phosphate buffers. So they are still used, but you just have to be aware of them. The choice of disaccharide. If we have a large molecule, we need to include an amorphous excipient in there. Typically, we use sucrose or trehalose. Maltose or lactose are available, but these are known as reducing sugars. What that means is that they interact with proteins when they're freeze-dried. In particular, they'll result in an interaction called the Maillard reaction that results in browning. That's what leads to meat browning when you cook it. So we stay away from those reducing sugars. We might add a crystallizing excipient. I often do because it makes it easier to freeze dry and more difficult to lead to failure. So you could add mannitol or glycine. In certain ratios, what cases for, I just had a question, what are the most used cases for mannitol and glycine? Mannitol and glycine, they don't offer any protection to your protein. And so what that means is that you cannot use them alone with the protein. You'll use them to form structure. So if my fingers are the mannitol and glycine, they'll make a nice scaffold so that they'll hold all the amorphous components in place, but still result in a really nice looking lyophilized solid. They also allow you to process at much higher temperatures, so it reduces your freeze drying time. So they add a huge advantage. But if you look at those marketed formulations, many of them don't include it. I've included them in the formulations I develop only because of that ease for drying. Good question. So for those of you not too familiar with freeze drying, this is a generalized process diagram. What we have is shelf temperature versus time. The red line is your shelf temperature. So we first have our freezing step. We cool it to some very low temperature, let's say -40, -50. Hold for a certain amount of time to freeze it. And then we could introduce an annealing step if we have a crystallizing component. That's where we would raise the temperature to, let's say, -15 or so, and hold for, let's say, 3 hours, just to ensure that it crystallizes. And then we can go into primary drying. That's where we initiate a vacuum, and then we remove that bulk ice, and we remove it based on a difference in vapor pressure of ice in the sublimation front and the vapor pressure of ice in the condenser. That's a whole nother lecture, so I'll just leave it at that. But at the end of primary drying, you should probably have around, let's say, 5% water, 2 to 5% water. And then we go into secondary drying. That's where we raise the shelf temperature. Pressure is no longer important here because we need energy now to remove the moisture that's what we call attached water, or water that can only be released through diffusion. And so we need energy there to remove it. And for that reason, we need to increase our shelf temperature, let's say to 30 or 40 degrees C, and hold it for a certain amount of time to reduce moisture. I'll show you later on where we conduct the studies to study the effect of residual moisture. And it goes into a little more in depth than this generalized diagram. So here are some basic questions to consider before we start formulation. First, this isn't a question, but we strive to keep things as simple as possible. So we only add components if we have data to demonstrate that they're needed. Our first question for proteins and monoclonal antibodies is, what is the effect of buffer type, buffer concentration, and pH on the stability of the ADC in solution? So we want to determine, well, is there an effect on buffer concentration? We don't want to go too high because high buffer concentration can cause pain upon injection, and it can also interact with your molecule. So we need to study a range of buffers in a range of pH and a range of concentration. Next is the ADC or protein sensitive to interfacial interactions. What we need to know is when we have our formulation added to our vial, do we lose protein or our ADC by it preferentially adhering to the glass surface? Or when we form ice, when we have all these ice crystals form, and my fingers are ice crystals, now we have a lot of contact of your molecule with the ice surface. It can then unfold and aggregate. Do we have that problem? So there are some studies that we need to do to conduct and determine if we need to add something such as a surfactant to prevent that. Finally, is our ADC sensitive to the method of transferring the solution from the tank through the filling needle into our vial? We need to think about how these formulations will be processed at full scale. And that's where these pumping studies come into play. First, this is the solution study I mentioned, where we look at the differences between types of buffer concentration, and pH. This sets up for a really nice statistical design of experiments to reduce the number of formulations we're preparing, reduce the number of samples we're sending to the lab, and reduce the amount of material we're using. So what we can do is formulate these and place them at 2 weeks, let's say at 1 to 2 weeks at 25 and 40 degrees C, and test them weekly. Here's an example of the data that we get from this. This is size exclusion chromatography, and we're looking at the statistical analysis after 2 weeks of storage at 40 C. First, let's look at buffer concentration. There was a slight effect. If you increased to 50 millimolar, so when compared to 10 and 30, we see more aggregation. And that's pretty common. You don't really need to go above, let's say, 20 to 30 millimolar in concentration of your buffers. Now let's look at buffer type. Here, histidine and phosphate seem to be much better with reducing aggregation than when compared to citrate. Next, pH. Decreasing the pH to 5 led to more aggregation. So now that helps us narrow in on our range of acceptable pH. We will also look at the ICE data. ICE data is telling us the same story. Basically, we want to stay between pH 6 and 7. We want to avoid the citrate buffer. In fact, histidine seems to be a bit better. And in general, with the ADCs that we've worked with here in our lab, most of them are quite stable and formulated well with a histidine buffer around pH 6. And then again, formulating between 10 and 30 millimolars. What this just helped us do is determine our center point. Now we can formulate at 15 millimolars of buffer, and we now have our acceptable formulation range. Now let's consider formulation development. So effective interfacial interactions. We typically need to design some type of study to look at the effective interfacial phenomena, particularly at the air solution interface, the glass solution interface, and the ice solution interface. So what we do is develop or create a shake study. And this doesn't have to be violent shaking. This is just shaking so that you constantly turn over the solution to expose the molecules to the air solution interface. And we'll set up something where we set samples prepared with and without a surfactant, and hopefully a range of surfactants, so that we know what the minimum amount of surfactant is needed to prevent aggregation. And we'll conduct this shaking for 30 to 60 minutes. And then we'll also do freeze thaw studies where we take the samples and freeze them and then thaw them for, let's say, 3 and 5 cycles. We often use polysorbate 80, but there's a mixture out there. I haven't seen any literature that suggests one is highly better than the other. Both are used to equal extent. And then we'll also look at a shear stress study, because a question comes up: do you use poloxamers as surfactants? They can be used. We haven't used them widely in our lab. I can't really tell you a strong reason why. It's just that we have a lot more experience with polysorbate. What surfactants other than PS20 and 80 have you seen commonly used, if any? The poloxamers, like what was mentioned. But again, we don't often use those, but they are out there. I'll show you a link to a website where you can look at where they're used and what types of formulations they're used and what concentrations. Very good questions. So let's just jump to the next slide. This is an example. This is an antibody-drug conjugate. These are actually from 2 different antibody-drug conjugates that were studied. Effective agitation and effect of freeze-thaw. And here we have polysorbate 80 presented at no. How do you mitigate polysorbate degradation? Oh, that's a good question. And you'll polysorbate degrading, is a challenge, because when they degrade, there can be, yes, they could form peroxides. They can even form peroxides in the bottle that you receive them in. So if you follow the manufacturer's instructions, they have especially Croda. If you work with Croda, they have specific instructions for how to store after you open the vial, because they're highly refined versions of polysorbate 80 do degrade. And so the storage conditions matter. And part of that storage condition is making sure there's a nice overlay of nitrogen in the vial that doesn't disappear over time. So with the amounts, the concentrations of polysorbate that we actually use in the formulation, I don't think I can name a time when I've seen a protein or a monoclonal antibody degrade as a result of polysorbate being present. But it's a good question. In these studies, we've prepared samples without polysorbate with a very low concentration. You can even go lower, 0.02%, but I've seen 0.01%. And then I made an artificially high one, 0.1%. And we look to see if we have any differences in the loss of main peak using SEC. And here we do not. And we do not have to include a surfactant in these formulations for the ADCs. This is the link here to the website. There's an inactive ingredient search that the FDA has. And it's a wonderful site. They will not tell you the specific product that it's used in, but it lists every product out on the market, whether it be topical, injectable, or oral, that has that particular excipient in it and at what concentrations. There's a question coming up, are ADCs more stable than classic monoclonal antibodies? From my experience, I'll have to say I lean toward yes. The antibodies we've worked with have been extremely stable, or ADCs we've worked with have been extremely stable, where there are certain monoclonal antibodies that show a far less stability. So yeah, it's a good question. And almost makes you feel like it's easier to work with, but don't let your guard down. You still have to perform these studies because they can still degrade. But just for your reference, check out that website. It really helps to show you what the maximum concentrations have been used. And there are, for some small molecule formulations that are delivered by IV infusion, the amount of polysorbate present is far larger than you would expect. So it's a good site to see what's out there. Here's a number of years ago, there was a conference where I can't remember who presented it, but there was a presentation there about the types of filling methods that were used. And someone had said that you should never use a piston pump. And I can't say that, but before I get into that, there's a question. Should we use polysorbates at their CMC concentration? By default, you already are. Most of these polysorbates have CMC concentrations that are very, very low. So you're automatically forming the CMCs. Back to the filling methods. So there are different filling methods in the manufacturing area. I'm only listing two of them here, but one is your peristaltic pump. That's the two pictures on your left side. That's where we use, let's say, most often we're using a silicone type of tubing that's threaded over those rotating bars, and they push, that's used as a positive displacement pump. So they rotate, and they squeeze and let go of the silicone tubing to transfer the solution. The big challenges with those is it's important to perform studies to determine how long can you use your silicone tubing in that pump before you start to see degradation of your silicone tubing. What we've seen is that there can be times for specific types of tubing, since they're being compressed over time, often they could split at the sides, or what's also very common is that the inside of the tubing rubs together and it creates spalling. So you'll end up with small pieces of silicone tubing in your filled vials. So those are studies that are important to conduct before you just use tubing for an extended amount of time. There's also what's known as the piston pump, and that's that lower picture on your left. I think I said left two pictures, but for peristaltic, I meant right. But the piston pump, what you're seeing there are two cylinders, and inside the cylinder is another cylinder that turns, that goes up and down, and it has to be narrow enough to fit into that outside cylinder without scraping. So that means there has to be a gap. That gap has to be filled, and it's filled with the solution that you're filling. So that means it's considered self-lubricating. What it does is that it moves, picks up a volume of solution, and then injects it into your vial. The concern here is that over time, since that solution is being self-lubricating, that your molecule will build up over time. Some of it remains trapped and is exposed to a lot of shear. For some molecules, that's a problem. For others, it's not at all. But it's important to conduct a study to determine if it is a challenge. So let's just look at one challenge. This is where we took solution and we recirculated it 5 times and 10 times through a peristaltic pump and through a piston pump. Why would I want to recirculate it? Well, I'm trying to imitate that solution being trapped in between the cylinder for the piston pump over time. And here we took those formulations and we placed them on short-term stability, 1 and 2 weeks. Why would we do that? Well, we learned the hard way. Before, we just wanted to know, well, do you see particles form over time, and just tested them at pre-pump and then T0. Didn't see any changes. And then we saw changes later on. So what that made us realize is that we need to place these on stability. And what we found is that with certain molecules, this is for one particular ADC, the piston pump over time was a bad choice because we saw a decrease or an increase in aggregation, a large decrease in main peak. So considerations for freeze drying a large molecule. I'm going to have to fast forward through here. I have a lot more slides here than I have time. We started a little bit late though. We need to realize that there's freeze concentration that occurs. And when freeze concentration occurs, that changes the environment in which your molecule is in. So that could lead to other changes. Some reasons why we'd want to add excipients. Well, if you have low solids content, you'll want to add something there for bulk. In general, since we're talking about an ADC, it's a large molecule, we have to have an amorphous excipient there. Amorphous excipients surround those molecules and prevent them from unfolding, maybe due to loss of water or from interacting with each other. So we can conduct a study to look at different sugar types. We've done this where we examine the effect of sucrose and trehalose while keeping mannitol constant. The reason why we do this is that if you're going to use a crystallizing component, they have to be at either the same concentration or more than your amorphous component. Otherwise, you risk them not crystallizing. So here's an example of a freeze dryer. It has a sample thief on there and on your product chamber. And then here's your condenser. So water flows from your product chamber to your condenser. Just as an overview, I've provided that before already, but so I won't go into depth here since I don't have much time. But just realize that there are different ways we could freeze things. The slower you freeze it, meaning if you choose to freeze at, let's say, 0.1 degrees C per minute, we see that there's a higher degree of supercooling. That means the solution does not freeze until very low temperatures. That creates very, very small ice crystals, and that could be a problem. We could also include controlled ice nucleation. This is an example from Millrock, where they use a freeze booster. That's where they're injecting a fog of ice into the chamber to act as seeds. So these solution vials are filled, or these vials are filled with solution and are partially stoppered or partially seated with stoppers that have an escape area. These happen to be single vent, but there can be dual vent. Oops, sorry. So this is an example of in-process data. What I'm wanting to show you here is the main portion of this is primary drying. And we can determine when primary drying is done by a couple of reasons. Again, that's a whole different lecture. We could look at product temperature. We could also look at reduction of pressure towards the end of primary drying. This is an example of stability for these freeze-dried solids. We found that, and this was time and again with the ADCs, they were more sensitive to pH than anything else. So formulating at this high pH 6.5 with trios, that was that formulation 8, over 2 months, we saw a large decrease in the stability. We're also concerned about, in your experience, how comparable is the lyophilization behavior of the ADC to behavior of the drug-free mAb? They're pretty much comparable because the monoclonal antibody itself, let's say, is about 150 kilodaltons. The ADC is very close to that. That linker system and the small molecule that's attached, they don't add much more weight. So they're very, very close. When we become concerned about, it's a good question, because what we're concerned about when let's say if we have to use a surrogate to do these studies, lyophilization studies, we might do that because we're concerned about the resistance to mass flow of water vapor from the solid to the condenser. And if your surrogate has a far different molecular weight than your antibody-drug conjugate, it could have less resistance to mass transfer than your antibody-drug conjugate, and therefore dry faster and easier. So those are some things to keep in mind. Let's say our goal here is that our dried solid occupies the same volume as your solution. The top we consider not acceptable. This was a formulation developed with just sucrose. And when you formulate with an amorphous component only, they tend to shrink. In this case, the temperature and also resistance mass transfer of water vapor were high. And so you resulted in much higher degrees of shrinking. You don't want that. In general, we want things to look good because on the market, you'll receive a lot of complaints if they have an odd appearance. They could be completely stable. In fact, in a lot of these collapsed formulations, we don't really see a large change in stability. It's just that they're difficult to reconstitute and they don't look pretty. And so here's some examples of poor appearance. This is collapse. This is partial collapse. And again, this was mostly an amorphous compound. And then this is an example of puffing. In this particular formulation, it contained glycine, and the original cycle did not have an annealing cycle. It was completely stable just like that. But if you add an annealing step, everything, the entire solid looks much more presentable. So those are just some considerations of why you want things to look good. I'm going to jump here. This is examples of, we want to conduct identification of failure studies. And that's where we use the same samples over and over again, let's say about 10 of them, and we freeze dry at warmer and warmer temperatures. And we want to look at the effective shelf temperature on product temperature and appearance. So I'm going to jump to this one. This formulation is formulated with mannitol and sucrose. And this is just an example of how stable it is during drying. The entire freeze drying cycle was conducted at 40 degrees C. Never did come close to the failure point, and the samples still looked presentable. A slight change, but that's just kind of creeping away from the wall. they were still perfectly acceptable. So the final point is we like to examine the effect of residual moisture on stability. And we do that by taking samples throughout secondary drying conditions and building a calibration curve using NIR and Carl Fisher. So we use a sample thief to start collecting samples throughout the end of primary drying and throughout secondary drying. So this is an example. Here, these steps are steps in shelf temperature. And this is how the residual moisture is affected by shelf temperature and the time at that shelf temperature. So let's hear at the end of primary drying at minus 25, this product had almost 4% moisture. But if you hold it at minus 10 for, say, a number of hours, moisture drops to around 2.5%, and so forth. And so what we'll do is we'll conduct a study where we're removing samples at specific conditions so that we have samples prepared with different levels of residual moisture. This tends to be a very consistent way of obtaining samples with definite residual moisture contents. We then take these samples, scan them using NIR to obtain the exact concentration of residual moisture at T0, and then store them on stability. And this is examples of how they change. This is T0 using DSC, and we see with high moisture concentration, the TG of this dried solid shifts to the left, so lower temperatures. That means possibly less stability. It even shifts after 2 months of storage even further. And another thing we look at is XRPD. X-ray powder diffraction is where we take these solids, we X-ray them, and they contain mannitol. Here I'm pointing out with the arrow mannitol hemihydrate. It tends to form in most conditions when you freeze dry it. And what we're worried about is the loss of that hydrate, because as it's lost, it releases moisture, and that can interact with your solids. In the cases where we've studied, luckily it hasn't been a problem. Our solids have still been, our molecules have still been completely stable, but it's something we still investigate. And then we, of course, include ICE, SEC. In this case, it's showing that with higher moisture, you're seeing a little more degradation. So I'll quit there because we've already gone pretty well past our time. I tried to answer questions as we went. Are there any additional questions that you could think of before we end? If not, I'd like to thank you for attending. Thank you for your attention, and hope it was useful to you. Brian has them, and Brian, as long as we can convert them to PDF, we can share the slides.
Sure. Thanks very much, Greg. There was one question here. I'm sorry I missed it, but you can explain again why a formulation with only sucrose can potentially result in cake shrinkage?
Sure. So these,
I don't know if that came out.
Yeah, it's perfect. These molecules such as sucrose and trehalose, when you freeze them and dry them, they remain amorphous. And when they remain amorphous, they have a lot of water associated with them. And so water acts as a plasticizer. So as you increase the temperature, even during your secondary drying, they can become slightly flexible. And when they flex, they shrink away from the walls of the vial. You'll always see some shrinkage. The goal is that you're not using such high temperatures that they shrink severely and don't provide a decent appearing solid.
Wow, Greg, that was just a real great webinar, getting lots of compliments here being shot out to us. Thank you so much, Greg. And everybody, enjoy your day. We'll send the slides out to you next week.
You bet. Thank you, Brian.
Thanks, Greg. Everybody, have a great, safe day. Bye.
Bye-bye.